Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 1H June 2019 paper. Figure 1 shows an animal cell viewed under a microscope. So we can see the animal cell, we can see the cell membrane. The cell contains a nucleus. What is the function of the nucleus? Now we know that a nucleus controls everything that is happening within the cell. So we're going to say nucleus controls the activities of the cell. Name one type of cell that does not contain a nucleus. We know that red blood cell do not contain nucleus. So we can write red blood cell. Now the main reason why red blood cell do not contain a nucleus is that red blood cell wants to increase the space in which the hemoglobin are carried, and also it wants to increase the surface area compared to its volume ratio. So to increase the surface area, it requires a biconcave disc-like structure, and to achieve that, what it does is that it dissolves its own nucleus. So in the beginning, a red blood cell is just like any other cell. It has a nucleus once it is born, and then once it produces enough amount of hemoglobin, and then it dissolves its own nucleus and then become that distinctive biconcave disc-like structure. The other cells that do not contain nucleus, you can write is bacteria or prokaryote and xylem. Draw a simple diagram of the cell in figure 1. Label two parts of the cell. When we are asked about the simple cell diagram, we will always need to consider that we are going to draw a cytoplasm along with, guys, avoid drawing a perfect circle. So we will draw a cell that closes on itself. We're going to draw a circular structure inside which is going to close on itself, which we'll refer to as nucleus, and then we can do some labeling. In this diagram, I will do several labeling, but you may only do two labeling because two parts of the label is needed. The other features that we are going to show will be mitochondria, and we will also show ribosome. So we are showing the cell membrane, and then we are showing cytoplasm. Then we can see ribosome, nucleus, and mitochondria. Guys, any two labelings are needed, so just label two. Name one structure found in plant cell but not found in animal cell. We can find chloroplast in plant cell. The other answers will be, we can find cellulose cell wall and permanent vacuole. These are the structures that are not found in animal cells. Figure 2 shows some different cells. We can see in figure 2, the line x to y is the distance measured for the cell. So we will measure it by a ruler. The real length from point x to y is 0.06 millimeter. Calculate the magnification. Use the equation magnification is equals to image size divided by real size of the object. In order to find this particular answer, we will first measure the distance from x to y, which in this case we are using a tab, so we will just take it from the mark scheme, which in this case is 2.4 centimeter. Since it is 2.4 centimeter, but this y, the original value, is given in millimeter, so we will write m is equals to size of the image. Instead of writing 2.4 centimeter, we will multiply it by 10, which will give us in millimeter, and the size of the real object, which is 0.06. Once we do the division, then we're going to get 400, which is the magnification. The cells shown in figure 2 were viewed using a light microscope. Give two advantages of using an electron microscope instead of a light microscope. Electron microscopes are able to give a very high magnification, so higher magnification. And the other advantage of using an electron microscope is that their resolution is very, very high, so higher resolution or resolving power. Guys, as an explanation for the resolving power, for those who do not know what is the resolving power, the ability to see between 2 points. You can see if I magnify these 2 points and I increase the image, you are able to see between these two objects as separate objects. You can see that they are separate objects. So this is, once we made it larger, that is magnification. The larger we make it, that is magnification. But once we make it larger, then we can see a clear dot. As you can see, these dots are very distinctively clear. So that is called resolution. Higher the resolution, the clearer you would be able to see. But imagine you have two dots that are very, very close. I'm going to draw two big dots, red dots, that are very close. As you can see, these dots are very, very close. Now, if I turn this page into its original shape, all right, and I make it smaller and smaller, you would probably at a point, let's say I'll draw the dots even closer to make my statement even better. All right, you can see the distance as I magnify it, you can understand it. But if I turn it into very small, as I make it smaller and smaller, it may seem to you that both of the dots are touching each other, while in reality they are not. You can see they're separate. They're distinctive dots. The resolution is the ability to resolve between 2 points. If you can resolve them as separate object, then you can say that it is a higher resolution. But if you can't say that they are separate object and they appear to be one, then you can say that is a lower resolution. Lower resolution will give blurry image at higher magnification, and higher resolution will give a clear image, all right, at higher magnification. Even though we're going to magnify the image, we'll still be able to see them distinctively separate. Mosquitoes carry a pathogen that causes malaria. What type of pathogen causes malaria? Now we know that malaria is caused by a type of organism known as protoctist. So protist will be the answer. And the other answers are not correct because malaria is not caused by bacteria, or neither it is a fungus, or neither it is a virus. Alright, so it is a protist, and the specific thing that causes malaria is called amoeba. It's an amoeba, alright, that causes the malaria. Mosquito nets can help prevent the spread of malaria. Table 1 shows the results of a study in one area of Africa. We can see number of people under the study, which is 476. Number of people who use mosquito nets when sleeping is 426. Who did not use mosquito nets when sleeping is 40, and who use mosquito nets when sleeping is 1.2. So you can see 40% of the people, those who slept without a mosquito net, so they were they were infected with malaria, and only 1.2% of people, those who were using mosquito net, were infected with malaria. Now a newspaper made the following statement: study shows mosquito nets are scientifically proven to prevent malaria. Give one piece of evidence that supports the statement. So we can definitely say that from this, all right, that lower percentage of people, those who were using mosquito nets, actually contracted malaria. So the answer will be lower percentage of people with malaria when using mosquito nets. Suggest one reason why the statement may not be valid. One of the reason the statement may not be valid is because even when using mosquito nets, some people got malaria. So some people who use mosquito nets have malaria. There are other options for the answer, and those options I will just mention to you as word. They are: the data is only driven from one part of Africa, so there is no peer review done on that particular data. All right. The size of the people, those who are used for that particular experiment, is very small, which is only 476 people, and only 50 of them did not use mosquito nets. So the group number is very uneven. All right, and there is no other information about people considered, for example, their age, their medical issues, the sickle cell anemia, or they taking anti-malarial drugs or not. So none of those were taken into account, and that's why this particular data can be invalid. Guys, all of those points that I have mentioned, I will mention them, all right, in the answers. However, this is the best answer, all right, that you can write, which is some people who use mosquito nets have malaria. Table 2 shows information about the number of deaths from malaria in the same area of Africa. Year 2005, number of deaths from malaria per 100,000 people is 161. And in year 2015, number of people death from malaria is per 100,000 people is 92. Predict the number of people over 100,000 per 100,000 who died from malaria in 2017 if the trend stayed the same. So since from 2013 to 2015, there is a decrease of only 2 people. All right. So we can consider the same for 2017. There will be a decrease of 2 people only, and we can conclude that the number will be 90. So 90 people. Use of mosquito nets has helped to reduce the number of deaths from malaria each year. Suggest one other reason for the reduced number of deaths from malaria each year. Now, since the question says that we have to suggest a reason, so we can give 3 reasons for this particular answer, and they are as the year progresses, we improve the healthcare within Africa and throughout the world in general. So since there is improved healthcare, so there will be cheaper treatments, cheaper vaccination, less people are likely to die. And then use of mosquito control methods, all right, for example, like using insecticides or using repellents, or in general people awareness, and the change in behavior to avoid being bitten by mosquitoes, knowing that mosquitoes can cause malaria in general population can also reduce the number of deaths from malaria each year. These are the 3 reasons listed in the mark scheme, so you can write any one of them and you'll still get a very nice, any one of them is a very nice point. Describe how the human body prevents pathogens from entering, defends itself against pathogen inside the body. So guys, when answering this particular question, we need to understand that this answer needs to be done it can be done in 2 levels. All right, in the level 2, all right, where we target for 4 to 6 marks, the answer should be scientifically with relevant facts and events or processes are identified and given in detail to form an accurate account of what the question is asking for. What are the question asking for? It is asking for prevent pathogen from entering and defend itself against pathogen inside the body. So first, our understanding will be how to prevent pathogens from entering. Now, to answer for the preventing of pathogens from entering the body, there can be many ways pathogens can enter. They are skin, stomach, eyes, and breathing system. So with the skin, we can say that it has a tough outer layer, and the skin acts as a barrier. The skin releases sebum on the surface of the skin. The oil repels pathogen, and scabs form over cuts, which acts as a physical barrier, and platelets are involved in the forming of the clots and scabs. So this is for the part of the skin. With the stomach, we can say the stomach contains hydrochloric acid. It kills bacteria in food the bacteria which is in the swallowed mucus from the airways. All of these are killed by the stomach hydrochloric acid. With the eyes, we can say that the eyes produces tears. It contains enzymes that kills bacteria. The tears are also antiseptic properties. We can talk about that. And for the breathing system, we can say trachea, nose produces mucus. The mucus is sticky, and the mucus traps bacteria, and the mucus is carried away by cilia, where it is swallowed into the stomach. And as we have already talked about it, it is killed by the hydrochloric acid. The bacteria are killed by the hydrochloric acid. Now, these are all physical barriers. Now, defending itself against pathogen which is inside the body, that means which has already entered inside the body, which is already in the system. How do we prevent it? So we have immune system, which are white blood cells. The white blood cells engulfs the pathogen, and antitoxins are produced. All right, antitoxins neutralizes the toxins. Antibodies are produced by white blood cell, which destroys the pathogen directly. And the white blood cell also produces memory cells, which gives a memory of that particular antigen, the foreign protein that we are receiving, and gives a more rapid response if the pathogen re-enters our body, and we do not get sick in that case. This question is about photosynthesis. Complete the word equation for the photosynthesis. So we know in photosynthesis we use carbon dioxide, which we then react with water in the presence of sunlight, and this produces glucose and oxygen. A student investigated photosynthesis using pondweed. Figure 3 shows the apparatus the student used. We can see the level of oxygen here accumulated in the measuring cylinder, and the student is using a type of pondweed which is under the light, and the condition provided is doing photosynthesis. Set up the apparatus as shown in figure 3. Switch on the lamp. After 20 minutes, record the volume of oxygen collected in the measuring cylinder. Repeat steps 1 to 3 using bulbs of different power output. That means the student is changing the light intensity and seeing the rate of photosynthesis. What was the independent variable in this investigation? So the power output of the bulb, all right, it is the independent variable. The rate of photosynthesis is the dependent variable. Time to collect oxygen, well, this is also a controlled variable because we will want a specific time to collect the oxygen, and the volume of the oxygen collected would be a dependent variable, which will then indicate what is the rate of the photosynthesis happening, the time and the volume of oxygen. So they are dependent variable, or they can be controlled variable, they can be in that range. So just 2 ways the method could be improved so that the results would be more valid. Any experiment can be made more valid when we repeat and calculate an average, when we control the temperature of the water, because the water temperature generally affects the rate of photosynthesis, and using different type of light bulbs can increase the temperature of the water. So we can use a water bath to maintain the constant temperature of the water. We can control the distance of the light bulb from the pondweed because the same light, when it is closer to the pondweed, will have a higher intensity of light, and when it is far away, then it will have a lower intensity of light. So we can control that. Then we can control the mass of the species or the age of the pondweed that is used, because it is important. If we use a different mass of pondweed in each experiment, then that mass will have a different number of leaves and thereby different ability to do photosynthesis. And then give the pondweed enough amount of time to equilibrate before actually performing the experiment, because once we bring in a particular light, a pondweed needs to do certain processes within itself before it can start to do photosynthesis by using that particular light. So give it enough amount of time to acclimatize. These are the points, guys. You only need to write 2. However, I'm going to mention you all of the 6 points. You can only choose from 2, which is the best from this 6. Table 3 shows the student's result. Power output of bulb in watts from 60 to 250 is used, and we can see the volume of oxygen collected in 20 minutes. We can see up to 100, and after 200 power output, the rate of the volume of oxygen collected in 20 minutes increases. However, from 200 and to 250, the volume of oxygen collected does not increase any further. So the rate doesn't increase any further, and we are asked to calculate the value of X in table 3. So because we are doing the CMQ cube per hour thing, and we need to know that 1 hour contains 60 minutes. When 60 minutes is divided by 20, it will produce the answer 3, which we will then multiply with the rate, which is 1.1 cm cube collected in 20 minutes definitely. So we will then multiply that, and that will give us 3.3 cm cube. So 3.3 cm cube per hour at 150 watts. Complete figure 4. You should label the x-axis, use suitable scale, and plot the data from table 3 and your answer to question number 3.4. Draw a line of best fit as well. So the rate of photosynthesis in cm cube per hour is given, and the x-axis is up to us. We have to do it. So first of all, we know that the x-axis is our independent variable. So x-axis will be power output of bulb in watts, and then we'll have to label up to 250. So we will do a labeling of this particular graph such that when we draw the graph, it will at least cover 2/3 of the graph paper. Determine the expected rate of photosynthesis with a bulb of power output 75 watt. So to do this, we will extrapolate at 75. So this is 75. We will draw a line touching our graph, and then we will extend that particular line to our y-axis, and our value comes out 1.85. So the rate of photosynthesis at 75 watt is likely to be 1.75 cm cube per hour. Guys, when answering a question like this, please do not be worried, because our answer will be checked based on our graph. So we can get it pretty much correct if we do it according to the graph. So the value was 1.85 according to our graph. So 1.85 cm cube per hour. Which graph shows the effective temperature on the rate of photosynthesis? So we can see the first graph where the temperature increases but the rate of photosynthesis does not increase. Photosynthesis is just like any other chemical reaction. As the temperature increases, the rate of photosynthesis increases. So this is also wrong, because here with increase in temperature, the rate of photosynthesis decreases. We can see as the temperature increases, the rate of photosynthesis increases up to a certain temperature. After that, the rate of photosynthesis decreases, which is typical of enzyme control reaction. As you know that as the temperature increases in an enzyme control reaction, once we exceed the optimum temperature of the enzyme becomes less and less effective, and at some point the enzyme becomes totally denatured, and as a result the enzyme doesn't work any further. So because of that, the rate of photosynthesis will definitely decrease. And in the last graph, why it is not correct, it's mainly because as the temperature increases, at first the rate of photosynthesis decreases. We know that with increase in temperature, the rate of photosynthesis should increase, so that is incorrect. And then again, with increase in temperature, the rate of photosynthesis increases exponentially, which is also wrong, because enzymes do not work at high temperature. So this is wrong as well. Water moves from a plant to the atmosphere through the leaves. How is the volume of water lost from the leaves controlled? Water is lost as water vapor from leaves. It is lost through the stomata mainly. Majority of it, at least 99.99% of it, is lost through the stomata. And the stomata, the plant can control the stomata, by opening and closing it, and it does it by using the guard cells. Describe the transport of water through a plant from the roots to the atmosphere. So water from the root is transported through the xylem to the aerial parts of the plant. And in the aerial parts of the plant, water evaporates from the leaves through the stomata into the atmosphere. A student investigated the volume of water lost from two plants of different species. Both plants were kept together. Figure 5 shows students' results. We can see. So plant A, the volume of water lost in cm cube is much greater than that of plant B, as we can see. And the time in hours in the experiment is also seen here. So just one reason for the difference in the rate of water loss from the two plants in the first 2.5 hours. So in the first 2.5 hours, why there is a difference? So it could be happening that plant A has more stomata and it is in a environment, all right, where there is plant, it is like, it has more supply of water from the root. So since it has more stomata, so more water is lost, whereas plant B has less number of stomata. So we can, because the question says suggest, so we can give few possible answers. One of them is plant A has more stomata, which is the best possible answer, I think. And then the other ones are plant A has more leaves, plant A has bigger leaves, plant A has a greater total surface area of leaves. Both plants were moved to a different place at 2.5 hours. Calculate the rate of water loss per hour in plant B from 2.5 to 3 hours. Give your answer to two significant figures. So we are asked to do it for plant B after 2.5 hours and up until 3 hours. So 2.5 hours to 3 hours, we can see that it loses about 2 to 6 points. So this is 7, and we can say that that is confidently 7.2. So we can see that the rate, right, we can extrapolate to find it out. So 7.2 minus 2, 7.2 minus 2 divided by 0.5 hours, which is equals to 10.4. So we can say 10.4 cm³ per hour. Suggest two reasons why the rate of water loss in both plants changed after 2.5 hours. So the rate increased because it is warmer, light intensity was higher, and the other added point that we can write is that the rate increased because it was less humid or it was more windier. Figure 6 shows the internal structure of the human heart. One of the heart valves is labeled. So we can see the valve. Sometimes a valve in the heart can start to leak. Explain why a person with a leaking heart valve has difficulty exercising. So a heart valve's function is to prevent the backflow of blood. However, with a leaking heart valve, the backflow can occur. So some blood can flow backwards. And since less blood leaves the heart, so less blood is pumped around the body instead of being pumped out, it stays within the heart. So less oxygenated blood is supplied to the muscle cells, and as a result less aerobic respiration takes place, so less energy is released and less efficient muscle contraction can occur. Due to the lack of oxygen, the person exercising will then do anaerobic respiration, and since anaerobic respiration is less efficient and the body is less efficient as also removing those buildup of lactic acid and the carbon dioxide, the lactic acid will then really build up and cause muscle fatigue. All right, and the person will be tired and will not be able to exercise. Guys, you will need to write only 4 points for this particular answer. However, you have to understand, to give a level 2 response you should refer to both respiration and the effects on the exercise. So if both of those respiration and the effect on the exercise are provided, you will have to mention 2 points for the respiration part and at least 2 points for the effect on the exercise, and then you will receive the 4 marks. A patient with a leaking heart valve may have the valve replaced. A study compared to 2 different types of replacement heart valve, mechanical valves and biological valves from pigs. The data used in the study was collected from female patients aged between 50 to 69. Number of patients given the valve. In the mechanical part, we got 2,852 and biological 1,754. Number of patients who died from heart-related problems after the replacement. In the case for mechanical, it's 180 and biological 178. So we can already see that the biological valve related deaths are higher in comparison to the number of transplants. Percentage of patients alive after 5 years. In mechanical, the percentage is higher than that of biological. Percentage of patients needing a second valve replacement within 6 years. In mechanical, it is lower. However, with biological, it is higher. Percentage of patients who had a blood clot on the brain after surgery. With a mechanical valve, it is higher, and biological, it is way lower. Give one conclusion about the death of patients from heart-related problems after a valve replacement. Include conclusions to support your calculations, to support your answer. So, first of all, deaths from mechanical valve is, about, we have to do the calculation. All right, that's from mechanical valve. All right, so it's 180 over 2852 times 100% gives us approximately 6.3%. And then we have to calculate deaths from biological valve, which was 178 from 1754, which is roughly 10.15%, or we can say 10%. Okay, so we can see therefore a higher proportion of patients die with biological valve. One risk of mechanical valve is that blood clots can form on the surface of the valve. Name the component of the blood that starts the process of blood clotting. Now we know that platelet is the first component that becomes sticky as as it encounters a cut in our body in the blood vessels. So we will name it platelets. In this case, the starting of the blood clotting is started with platelets. Evaluate the use of mechanical replacement valve and biological replacement valve, heart valves. So when asked a question like this, we have to think of level 1, level 2, and level 3. Level 3 being the highest, which will have between 5 to 6 answers. Marking points, and it will give you 5 to 6 marks. So in level 3, a judgment strongly linked and logically supported by a sufficient range of correct reasons are given in these cases, because we have to evaluate from a particular given data. So how we can write this particular answer? We can first of all say mechanical valves. Let's see the conclusion from the table here. Mechanical valves, as in general, requires less replacement, means it is long lasting. However, it can cause blood clots after the surgery. So the patient will need to take anti-clotting medication for the rest of their life until they are alive. If medication is not taken correctly, then blood clot can form, which can be a problem. All right. Medication, since it is for, not clotting, then if there is a cut or bleeding, then the person will suffer from excessive bleeding. Some patients say that they can hear the valves opening and closing as well. Survival rate after 5 years, all right, is very, very high. After 5 years is very, very high in comparison that of the biological valves. Lower percentage of death due to heart-related problems. In biological valves, we see the person does not require any additional medication. However, there can be ethical issues using, surrounding the use of animal tissue, and there can be a problem because it is a biological material. The valve can get calcified and it can harden. So it's more likely to need further operations to replace the valve, and there can be, a likelihood of rejection. And to prevent the rejection, then the person may need to take immunosuppressant drug for the rest of their life, which can compromise their immunity. Now the thing is, the best thing about both of these valves are they are readily available to the patients, and there is a very little to no waiting time to get it, so which is a very big advantage for the patients as well. Guys, I will give you a good indicative answer, and you can choose the points from there which ones you want to write, but you have to write in a logical sense, including at least 3 points for the mechanical valve, 3 points from the biological valve, and 1 point which will indicate for both of the valve. This will definitely ensure that you will have a level 3 response, okay, and you will get definitely full marks. People with diabetes have difficulty controlling their blood glucose concentration. Which part of the blood transports glucose? Now the question say the answers are it says lymphocytes. We know lymphocytes are that type of white blood cell, and they are needed for the immunity. Plasma is the straw-colored liquid that carries many dissolved substances, including glucose. Platelets are they're fragments of cells that are needed for blood clotting, and red blood cells are cells. They do not contain nucleus, but they contain hemoglobin for the carrying of oxygen. So plasma will be the answer. Glucose is often found in the urine of people with diabetes. Name a chemical used to test for glucose. In the school laboratory, we can use Benedict's reagent for testing glucose. Describe a test that could be used to show that the person's urine contains glucose. A test. So we can add Benedict's solution to the urine and we can heat. A positive result will be an indication of blue color, which will change to brick red. The body cells of a person with untreated diabetes lose more water than the body cells of a person who does not have diabetes. Explain how diabetes can cause the body cells to lose more water. Since diabetes causes a high blood glucose concentration, the more concentrated the blood, then the solution that are in of the cells, then water moves out of the cells, all right, into the blood by osmosis. And as a result, as the water moves through the partially permeable membrane of the cell, they move into the blood, and this dehydrates that particular person at a cellular level. Glucose is absorbed into the blood in the small intestine by both diffusion and active transport. Describe how the small intestine is adapted for efficient absorption. So, first of all, we will talk about the structures of the small intestine that gives it a better advantage. First of all, the projections, or we can say, the folds or the villi, provide a large surface area. The walls of the projections or folds or the villi, they have capillaries, which are, only 1 cell thick for very short diffusion. diffusion distance. So the small intestine is also very long, which increases the time for the absorption of the glucose can occur, and it contains a very good supply of blood to maintain a higher concentration gradient, between the blood that is flowing through the stomach, through the gut system, and the small intestine. So as a result, all right, these cells, they can absorb more glucose. The blood can get more glucose through the small intestine. Now the cells in those lining, in those projections such as villi, have mitochondria which can do aerobic respiration, and then they can do active transport of the glucose as well. A small animal called an axolotl lives in water. The axolotl has a double circulatory system. Define the term double circulatory system. So when we talk about double circulatory system, we have to first of all understand the meaning of the word double. The word double means the blood is pumped to the lungs by one side of the heart, and the blood is pumped to the body by the other side of the heart. An alternative answer for this particular question would be blood enters the heart twice for every one circuit around the body. Figure 7 shows the double circulatory system of the axolotl. Alright, so the diagram is actually not represented because of third-party copyright restrictions. However, I'll give you a picture. Just give me a moment. The heart of the axolotl has only one ventricle. However, guys, in this particular image, we are not showing the heart of the axolotl. Alright, we are showing the heart of a human because the original picture was removed due to third-party copyright restrictions, so we cannot use that particular image. So anyways, the question asks us label the ventricle on figure 7. So we can label the ventricle on this particular figure, where this particular part is the ventricle. Explain why having only one ventricle makes the circulatory system less efficient than having two ventricles. So basically, oxygenated and deoxygenated blood are able to mix if there is only one ventricle. So less oxygen or oxygenated blood is able to reach the body tissues. Figure 8 shows an axolotl. Explain why an axolotl may die in water with a low concentration of oxygen. So our key point here is that the axolotl will die and there is a low concentration of oxygen in the water that it is living. So if the axolotl is living in a low concentration of oxygen containing water, the concentration gradient of the oxygen is less steep. Therefore, less oxygen will be able to diffuse into the blood of the axolotl through its gills. So less aerobic respiration can take place in its body, and as a result the metabolism decreases, and the axolotl slowly, due to the anaerobic respiration that takes place within its body, produces lactic acid, which is toxic, and the lactic acid will then kill the axolotl. If a gill of an axolotl is removed, a new gill will grow in its place. Scientists hope to use information on how axolotl grow new gills to help with regenerating human tissue. Name the type of cell that divides when a new gill grows. So, to grow a new gill, the axolotl may need to divide its stem cells. Name one condition that could be treated using regenerated human tissue. Regenerated human tissue can easily, treat diseases like paralysis, diabetes. Suggest one reason why an axolotl is suitable animal for research in the laboratory. So one of the reasons axolotl or any animals in general will be suitable for research in laboratory, it's because they're easy to breed, they're very cheap to keep and rare, they're small, relatively small, and do not take up a lot of space. Any one of these possible points will be the answer, so just feel free and write one of them. An axolotl may not be suitable animal to study when researching regeneration in human tissues. Suggest one reason why. One of the main reasons that we can come up with, because it is not a mammal, rather it is an amphibian. The other possible answers are regeneration in gills may be different to that in other organs or other parts of the body. And finally, the body or the metabolism of the axolotl are too different compared to that of humans. Any one of these points are possible answers. So just choose one of them and feel free to write it whenever you get a question like this. Pancreatic cancer develops when a malignant tumor grows inside the pancreas. The pancreas produces digestive enzymes. What is an enzyme? An enzyme is a chemical which catalyzes or speeds up a reaction in living organisms. We can also write a chemical which catalyzes or speeds up a biological reaction. We can call it an enzyme. The simplest of answer that we can give is like this. An enzyme is a protein which speeds up biological biological chemical reaction. So to get the 2 marks, we'll have to write enzyme is a protein, and then we'll have to mention any one of those other points. The fact that we write that an enzyme is a protein gives us the first mark. Carbohydrates is an enzyme produced by pancreas. Name 2 other organs in the digestive system that produce carbohydrates. Carbohydrates is produced by salivary gland in the small intestine. So it is either produced in the salivary gland or in the small intestine. One symptom of pancreatic cancer is weight loss. Explain how pancreatic cancer may cause a person to lose weight. Do not refer to hormones in your answer. Person with pancreatic cancer will not produce or will produce very little amount of enzymes from the pancreas. As a result, food is not broken down fully or digested fully. So less glucose is absorbed from food into the bloodstream, and less glucose is available for respiration in the body. As a result, the person use up the storage of fat for metabolism. Fewer amino acid is also absorbed from the food, and as a result, very less amino acid is available for making new proteins. Fewer fatty acid is absorbed from the food. As a result, fewer fatty acid is available for storing fat in the body or making new cell membrane. And a person with pancreatic cancer may undergo chemotherapy, and which will then lead to loss of appetite. All right, and the person may take less food in general. Guys, I'll mention the points, those that are obligatory to write, and I will also mention the points, those that you can choose from. So guys, you have to mention the mandatory points, and after mentioning the mandatory points, you can choose any one of the pairs from the or groups, and you will get a full marks for your four marking points. Enzymes A and B are involved in controlling cell division in pancreatic cancer cells. Most cancer cells produce both enzyme A and B, enzyme B. Some people have a gene mutation that stops cancer cells producing enzyme B. Figure 9 shows how cell division is controlled in pancreatic cancer cells. So we can see enzymes A or enzyme B. Cells divide uncontrollably. Pancreatic cancer cells with no enzyme cells stop dividing and are destroyed. Scientists have developed a drug that inhibits enzyme A. The drug is given to the pancreatic cancer patients who have the gene mutation that stops cancer cells producing enzyme B. The drug only targets cancer cells. Explain why the drug can be used to treat pancreatic cancer in patients with the gene mutation. So basically, the understanding of this particular question is this. If we look into this particular person, let's say the first example of the particular person is enzyme A and B both are being produced. If enzyme A and B both are being produced, the cell will divide uncontrollably and there is no control of it. If none of the enzymes are produced, then the cell stops dividing and those cancer cells are destroyed. Now, if a particular person already has a gene mutation that causes enzyme B, to be produced, then enzyme B is already eliminated. All the doctors need to do is give the patient a medicine to prevent the pancreatic cells, cancer cells, from producing enzyme A, and the uncontrollable cell division will be stopped. So that's why now the question says, explain why the drug can be used to treat pancreatic cancer in patients with gene mutation. So we will say the answer that can be written is like this. The person with the gene mutation already does not produce enzyme B. We are giving the particular person drug to inhibit enzyme A, then the cancer cells will not be able to divide and will be destroyed. So the tumor will not be able to grow any bigger, and they are very unlikely to produce a secondary tumor because enzymes A and B both are not working and they are inhibited. Explain why the drug cannot be used to treat pancreatic cancers in patients that produces both enzyme A and B. The drug only inhibits the enzyme A. So if the enzyme A is inhibited and the person still produces enzyme A and enzyme B will still be present in that particular person. So the answer can be written like this. The functional enzyme B would still be present. Therefore, cancer cells will still be able to divide uncontrollably, and that will be our answer. The drug was trialed before it was licensed for use. To improve the validity of the results in the trial, some patients were given a placebo. A double-blind trial was used. Give reason why a placebo and a double-blind trial was used. So the purpose of the placebo. A placebo is given to avoid the patients thinking they feel better with the drug, or to take into the account of a psychological effect of taking a drug, because sometimes a patient may just get better because they are taking placebo. So we can also say that it is acting as a control or comparison, whereas a double-blind trial is actually done to avoid a biased result. One stage in a drug trial is to test the drug on healthy volunteers. What is the next stage in the drug trial? So once the drug is trialed on healthy volunteers, we are mainly looking for side effects. Now, if the side effects are proven to be manageable or controlled, then we can test on volunteers with disease. Let's see the other options. Testing on all patients with the disease. Now, we are not going to test it on all patients because they did not provide their consent. Testing on human tissue. Testing on human tissue is done before testing on healthy volunteers. Then testing on live animals. Testing on live animals is also done before testing on healthy volunteers. A monoclonal antibody has been produced to treat pancreatic cancer. Explain how the monoclonal antibody works to treat pancreatic cancer. A monoclonal antibody is just, first of all, we need to understand that this is an antibody that is against a particular protein. So monoclonal antibody, what it can do is we can attach it to a radioactive substance or a toxin. Because this particular antibody is very specific, all right, the monoclonal antibody will then only attach to the target antigen on the cancer cells, and the radioactive substance or the drug or the toxin will bind to that particular cell and stop them from growing or dividing. Guys, that's the end of this particular question paper. Thank you for watching the video, and guys, subscribe to the channel, and let your friends know question paper like videos like these are uploaded daily. Let the channels grow, all right, and write in the comments that what kind of question, how you want us to actually solve the future questions, and let us know in the comment in general about your opinion, and we will definitely try to improve on that. We do record a lot of question paper earlier on and then basically publish them one by one, so your whatever you suggest will not appear just right in the next video. However, we will definitely we read all the comments, and we definitely take your comments in the mind and basically try to improve on that. So yeah, guys, thank you for watching the video, and best of luck with your exam. See you in the next video, guys. Bye bye.